Well, thank you for inviting me back. Give an update on N-acetyl-L-leucine in flies and Syngap 1. I just want to make a comment. I noticed this today that the color scheme on my lab's logo is nearly identical to the color scheme on Syngap's logos. And I made this logo before I met Mike and Syngap. So you knew the partnership was match made in heaven, right? So, all right. So a lot of this work, oh, look, we were wrong about the slides. It's messed up too. But a lot of this work is brought to you by repurposing, and I'm not gonna go through that again. But kind of finding new uses for old drugs, right? And trying to dig into that treasure chest of unknown biology and medicine. And also by this super annoying pest that you have flying over your bananas while you're sitting here at home. Drosophila fruit fly, it's a pest in your kitchen, but is an incredible biomedical tool and allows us to work at a speed that sorry, mouse people, that you can't work at. But it's really allowed us to kind of make the kind of progress that we've been able to make. And all this work was done by an incredible team that I have in my lab who helps run drug screens for rare disease foundations like SRF and other groups who are maybe here in the audience as well. So last year I told you about this eye-based Syngap 1 drug screen that we generated, and kind of the beauty of having flies is that we can generate a model like this where we eliminate Syngap function in the fly eye, and it gives you this really gnarly phenotype on the right compared to the kind of control healthy eye on the left. And this is the basis of our screen. We can measure it, and you can see that it's quite a bit smaller than the control wild type eye, and the screen is really simple. We simply ask, can a drug move this yellow box up towards the purple box? And that would be considered a biological rescue. Oh, I'm missing slides in here. There are a couple slides that are not in here. All right, well, I'll tell you about it anyways. So we ran this in 1,500 drugs, FDA approved drugs. We collected eye images from all those 1,500 drug treatments and measured them and generated a score for each of those drugs. And we got a list of about, I don't have my slides, so I can't remember the number anymore, but about 30 really exciting. So we got about a list of about, I think, 30 drugs that had a positive hit on this eye phenotype. So it moved the yellow box towards the purple box, and I talked about some of those hits last year and kind of the categories that we found. Skipping 4 slides that I'm missing from my talk. We settled on N-acetyl-L-leucine as one particularly interesting hit, and one reason this was interesting was just kind of the safety profile of it, right? N-acetyl-L-leucine is an amino acid, and so we kind of ran with this. One of the slides that's missing here makes the point that N-acetyl-L-leucine is actually not even in the top 10 hits of our screen, but still ended up being pretty interesting. And so that just kind of speaks to the nature of using these kinds of tools to find candidates, but don't treat it as gospel. What happens in humans might be very different than what happens in our models. So we validated this in many different ways now, but this is just one example where we can do a dose response, and there isn't actually much of a dose response. It seems to have a pretty strong effect no matter what dose we use, but you can see that it improves the eye size across the different doses. Ooh, this is not right. And so this is what I'm missing, like 4 slides in between these slides. But so N-acetyl-L-leucine is this molecule you see here, and again, it's a modified form of L-leucine, which leucine is just an amino acid that you use in your body for all kinds of processes, and it's basically just the addition of this acetyl group onto the leucine molecule. And it didn't come out of nowhere, as you may know, it's been tested for a number of different neurodevelopmental and neurodegenerative disorders in the past few years as a potential therapy, and as was just mentioned, was recently FDA approved for intravial use as Acnersa. Many of you know this as Tanganil, which is actually a mixture of two different forms of acetyleucine, N-acetyl-L-leucine and N-acetyl-D-leucine. So L is on the left and D is on the right. You can see that they're slightly different. The acetyl group sticks out of the page in one and into the page in the other. So they are different. It's subtle, but it's a big difference. And again, I don't have to say this to many of the families in the room, but it seems to be having a positive effect in Syngap individuals. This is from the SRF Web's blog, where they've highlighted one of the examples and kind of the different measures that have been tracked, and highlighted green are things that have improved, and highlighted red are things that have not improved or gotten worse. And as you can see, almost everything has showed some kind of improvement, which is really exciting. When we started doing this work, we thought, well, maybe it's even easier than N-acetyl-L-leucine. Maybe it's just leucine, right? And maybe the body just needs leucine to do this. But it turns out that when we tested other forms of leucine, none of them work. So on the left is N-acetyl-D-leucine, the other half of Tanganil, and it doesn't work on its own. L-leucine on its own doesn't work. And this other molecule, for which I cannot pronounce, is the direct metabolite of L-leucine, and that doesn't work. And so there's something really special about N-acetyl-L-leucine that's kind of driving its potential. And it turns out we probably already know this from the literature. N-acetyl-L-leucine is transported in the cell by this transporter called MCT1. And it turns out this acetyl group is the big difference. L-leucine is transported by a completely different transporter and very tightly regulated, so that once the cell has reached whatever it needs, you don't get any more L-leucine into the cell. But by adding the acetyl group to L-leucine, N-acetyl-L-leucine now goes through this other transporter for which there is no checked transport, and it just floods into the cell. And as soon as it floods into the cell, the acetyl group gets cut off and it gets converted to L-leucine within minutes and hours of taking it. This happens in humans and mice. And so we think that N-acetyl-L-leucine is actually just a prodrug for L-leucine. A prodrug is a molecule you take. It's not the active form. Your body actually converts it into the active form. And we think that N-acetyl-L-leucine is a prodrug for L-leucine. And that's why adding leucine to the diet doesn't work, because the body's probably already at whatever it needs. And it's tightly regulated, and kind of adding it into the diet doesn't really make a big difference. And so what is it doing biologically? It turns out L-leucine can have a number of different effects. You can imagine it's an amino acid, so it can impact anything that's protein translation related, but it also impacts glucose and energy metabolism, which we'll get back into a second. It has really strong effects on mTOR and autophagy. Autophagy is kind of like the cleanup crew of the cell. And other pathways as well. And these are things we're going to dig into in the next year. And so the next steps for our lab is to really ask these two questions. And actually, I meant to put a third question on here, which I'll touch on at the end. But can N-acetyl-L-leucine rescue other SYNGAP1 phenotypes? So we're starting to now expand beyond this kind of weird eye phenotype I showed you to something a little bit more relevant to the patients. And also, how does it work, right? Like, if we can figure out how N-acetyl-L-leucine works, there might actually be a better drug, right? That targets the actual pathway for which N-acetyl-L-leucine is impacting. And that might be even better than kind of taking this kind of modified amino acid. And so the first thing we've been asking is, can N-acetyl-L-leucine rescue other SYNGAP1 phenotypes? And we've just begun this work, and a lot of this work is the work of Haley Tokars, which is a grad student in my lab. She's sitting in the back. You can ask her questions at the end. But we know that in the fly, complete loss of SYNGAP1 is lethal, just like in mice and probably in humans as well. Interestingly, loss of SYNGAP1 in the support cells of the nervous system glia is also lethal. So these are, again, the cells that help the neurons function properly. And if you eliminate SYNGAP in those cells, the flies don't live. And if you eliminate Syngap in all neurons, it results in a movement disorder and also a seizure disorder, which is quite similar to what we see in many of the patients. And so I'll show you a little bit of this. So it turns out flies have seizures. Um, People think this is weird, but flies have seizures, and they actually go through a pretty similar seizure profile as you can see on this diagram. It looks very similar to what we see in our patients. And in flies, the way we measure seizures is we often have to induce them, and so we put the flies in a vial and we vortex it at about 10,000 RPMs. It's really weird. We vortex it for about 10,000 RPMs for 10 seconds. Healthy, normal flies will get up like you did nothing to them. They'll just get up and walk like nothing happened. Flies that are susceptible to seizures, and there are all kinds of models that are now, will go through this phase that you see here in this diagram. So I'm going to show you this here. So on the left is a wild type control fly. Can you click on that for me? Oh, it's not. Yeah, there it is. So you can see this was right after they vortex and they just got up and climbed up the top. Climbed up to the top. Now, please click on the next one. This is vortex, put them down, and you can see that those are the Syngap flies, and they are not like the flies on the left, right? They are some of them are undergoing seizures on the bottom, and if I were to rate the seizure phenotype that we see in these Syngap flies, it's actually fairly mild compared to other flies we have. Other flies we have will completely pass out for minutes and not even move at all. And these guys are not definitely not normal, but also not incredibly severe either. So one of the things moving forward is we want to ask, can NALL rescue these phenotypes? So we can redo all this analysis now in the presence and absence of NALL and see if we see an improvement. Other things that we're going to do as well is eliminate Syngap function in subsets of different kinds of glia and subsets of different kinds of neurons. So we can eliminate just in excitatory neurons or just inhibitory neurons, just in dopaminergic neurons or just in serotonergic neurons or inhibitory neurons, and see kind of where the phenotype is strongest and also where NAL has the strongest effect. So these are things that we can do fairly easily with flies. So next we want to know how it's working, and these are all kind of things we're going to dig into. One of the ways we've kind of started getting at this is actually using RNA-seq. This is just looking at gene expression, and in particular we were interested in asking how is RNA-seq affected in how's the RNA-seq affected when we give the Syngap flies NALL? What is changed by this drug? And so we kind of had this four group design where we have healthy flies with no drug, healthy flies with NALL, Syngap flies without drug, and Syngap with N-acetyl-L-leucine. And I'm just going to highlight this. Oh, these slides are interesting. We're going to highlight this this one particular comparison where we compared Syngap flies only with or without N-acetyl-L-leucine. And there's lots of interesting things in this data, and some of it's on Haley's poster. But one of the really interesting things we found was that amongst the genes that are downregulated by N-acetyl-L-leucine in the Syngap flies is a huge enrichment for tyrosine catabolic process. Now, tyrosine is actually another amino acid, which is really interesting here, and catabolic process is the breakdown of tyrosine, and so this is what is a snapshot of what's happening in our RNA-seq data. So this is the pathway for tyrosine breakdown, and actually tyrosine gets broken down into components that actually get fed into energy metabolism. So you remember energy metabolism was one of the points I brought up in the other slide. And you can see in red a number of the enzymes that are involved in this process are actually downregulated when we give them N-acetyl-L-leucine. I don't know why. I mean, it's possible that there's too much energy production, but it's also possible that maybe tyrosine needs to be shunted to dopamine. For example, dopamine tyrosine is a precursor to dopamine, and if you shunt the pathway, if you shut down the pathway in one direction, it'll just get pushed in another direction, right? And so these are hypotheses that we can test fairly easily in flies, kind of using some of the neuron-specific knockdowns that we talked about earlier. So again, work in progress, and stay tuned for what this means. But kind of as we move forward, um, this drug repurposing has given us a potential molecule that's probably having some positive therapeutic benefit to some of the SYNGAP individuals. And one of the things that I meant to kind of put as a third point was that we want to try to understand what drives differences in response to a drug like NALL and acetyl-L-leucine. We've heard from some families that it sometimes helps with the seizure, sometimes it doesn't, and some families don't seem to have response at all. And one of the things we can do in flies is we can take our SYNGAP model now and cross it into a couple hundred different genetic backgrounds. So rather than studying a single SYNGAP patient, we can study a couple hundred SYNGAP patients in the flies. And then we can ask, well, how does the SYNGAP phenotype vary across those hundreds of different fly strains, which is what we have in our community. And then we can ask, how does NAL affect all those different backgrounds? And then map genetic variants that drives different responses to NAL and SYNGAP loss. And so that in that way we can start doing this kind of pharmacogenomic approach of trying to identify which variants or which patients might have a better or worse response to a particular drug like an acetyl-L-leucine. So with that, I will end and thank people in my lab and of course the support of SRF for this work.